we are essentially interested in developing gene therapy for HIV infection. As you know, CCR5 provides a wonderful target for gene therapy because of several reasons. One of the things is that the CCR5 is the major co-receptor for HIV, and people with naturally occurring homozygous CCR5 mutation, the delta 32 mutation, which occurs in 0.1 to 1% of the population, they are highly protected against HIV. They do not acquire HIV infection. And even heterozygous mutation also confers partial protection. And the so-called Berlin patient, who is the one and only patient actually cured of HIV, here he was a patient with HIV and lymphoma, and he was being treated for lymphoma. So after myeloablation, he underwent a bone marrow transplant using CCR5 minus donor. So this, he completely cured HIV in this person. And it's been over 8 years now. And CCR5 deficiency can also be induced by several means, like RNA interference, gene editing. In animal models, it's been shown to confer HIV resistance. Therefore, CCR5 gene disruption provides a very good option. So what is gene editing? In theory, the HIV-infected person can be transplanted with bone marrow from homozygous CCR5 delta 32 individuals. However, it's very difficult to find such HLA-matched donors because the mutation occurs in 0.1 to 1% of the Caucasian population, much lesser in the others. So the more practical way is to create CCR5 mutation in the patient's own cells. That's what exactly is gene editing. And newer gene editing technologies can be used to create mutation. And these are some of the new methods for gene editing using zinc finger nuclease, TALEN, CRISPR-Cas9. And we have used zinc finger and CRISPR system for this. So what are zinc finger nucleases? These are essentially chimeric protein. It contains, there are transcription factors called zinc finger transcription factors, which has two components, one for DNA binding, the other for activation domain, activating the gene expression. So here you take just the DNA binding component of the zinc finger protein and fuse it to the FOC1 endonuclease. So this is what is considered the zinc finger nucleus. And actually, each module of the zinc finger nucleus consists of about 30 amino acids and recognizes only 3 specific nucleotides. So generally, you have 4 to 6 modules linked together so that you can recognize about 12 to 18 base pairs. And the other aspect is that the endonuclease, FOC1 endonuclease, has to dimerize for it to induce DNA break. So therefore, typically what you do is you construct 2 arms of the zinc finger nucleus, the left arm and the right arm, targeting about 12 to 18 nucleotides on either side of the DNA, where you want to induce a break with about 6 base pair loop. So what happens is that once the zinc finger nucleases come together, the FOC1 endonuclease dimerizes and it cuts the DNA within the spacer region here. And once the DNA double-stranded break occurs, it is repaired by generally the error-prone system called the non-homologous end joining system. And this error-prone repair induces deletions or insertions and resulting in disruption of the reading frame and gene expression. The way most commonly used to express CFNs is to clone it in a lentiviral vector. Lentiviral vector here, for example, the right arm and the left arm of the zinc finger nucleus linked with a protease cleavage domain P2A, and it's expressed in a self-inactivating lentiviral vector. and you transfect essentially 293 T cells with this plasmid along with helper plasmids. You have to provide GAG-Pol, Rev, and pseudotype generally with VSVG. That's vesicular stomatitis viral glycoprotein. So this results in integration of the lentivirus and long-term expression of the ZFNs. But the problem with this approach is that one of the major problem with nuclease is off-target effects. In addition to CCR5, it can cut some other targets that you don't want to. So the off-target effect increases with nucleus exposure. Longer the nucleus exposure, the off-target effect is more. And in the integrating lentiviral vector I showed you before, the integration of the vector results in long-term expression of ZFN. And also, the commonly used VSV-G pseudotyping allows lentivirus translation only in activated T cells, not in resting T cells. So to overcome these two things, we first of all use the lentiviral vector, which is non-integrating, which has been mutated in the integrase. So we cloned the CCR5 in this vector and pseudotyped using, instead of VSV-G, HIV envelope. The idea here is that the lentivirus then transduces only HIV-susceptible cells, including resting T cells, and resulting in short-term exposure of the nucleus. So if you look at the expression, so the lentiviral vector also contains GFP, so you can monitor expression via looking for GFP expression. And whereas this is the integrating lentiviral vector, the expression continues up to 10 days and beyond. Whereas if we use the non-integrating lentiviral vector, the expression goes away very soon, day 6 onwards. So we use this system to transduce kidney embryonic stem cells, 293 T cells or Jurkat cells. As you can see, this is the transduction efficiency. And gene editing we can detect by PCR amplifying the CCR5 region and using what is called the surveyor nuclease assay or cell-1 assay. So corresponding to this, you can see about 80% gene editing in this cell and about 35% gene editing in the Jurkat T cells. So next, we wanted to test whether it also works in primary T cells. These are primary human T cells, CD4 T cells. And this is the transduction efficiency with the vector or with our ZFN. So whereas if you look at the gene editing, it only occurs when with these two, in the resting as well as activated T cells, about 22 to 28% gene editing you can find in primary T cells. So now the question is whether we can use it to resist HIV infection. So first we tested in vitro. CD4 T cells are activated, transduced with ZFN, and then infected with HIV. So if you do that, you can see that these are the controls, and you can see reduction in HIV titer by over 2 logs. And the activated as well as in the resting T cell. This is the control, whereas this is the CCR5 modified T cells. Now the question is whether it also can be tested in vivo. For this, we use the mouse model. These are heavily immunodeficient, not SCID, IL-2 receptor gamma chain knockout mice. These mice don't have their own T cells or NK cells or B cells. So now you can put human cells into these mice, and the human cells grow because of xenogenic activation. So this provides a convenient model to test the effect of gene therapy or drugs on HIV infection. So essentially, this is the strategy here. First, we isolate human CD4 T cells, transduce with non-integrating lentiviral vector encoding CCR5-ZFN, and inject into mice. Check for human T cell engraftment, and then infect with HIV. And then follow up for CD4 depletion, and at the last time point, we sacrifice the mice and do viral titers. So if we do that, you can see this is the CD4 T cells numbers. So in the controls, the CD4 T cells decline. For example, this is the starting number, and because of HIV-induced deletion, the CD4 T cells are coming down, whereas in the CCR5 treated mice, the loss is much less. And in fact, over time, the CD4 counts improve. And if you look at the viral titers, we have here the P24 assay, and compared to the controls, it is much reduced, and even by the QPCRs, the viral titers are reduced compared to controls. And this is the percent gene editing in the cells isolated from the mice. So what this shows is that CCR5 gene modification confers protection against HIV infection. But next we wanted to know what happens if we treat the cells from patients who have been already infected with HIV, whether we can continue to give protection. So here there are 3 patients. This patient was treated with antiretroviral drugs, and these 2 patients are not treated with antiretroviral drugs. And again here, if you look at the CD4 T cell levels, this is the ZFN, CCR5 disruption, this is the control. So over time, and each dot is a mouse. Means we have about 5 to 6 mice per group, and you see that the CD4 T cell decline is much less in the treated versus controls. And the viral load is much lower in the CFN treated versus control by 2 assays. And interestingly, these are patients who have not been treated with antiretroviral drugs. You can see that these CD4 levels are already low to begin with, and it goes down further with HIV infection in the controls. Now, if you treat with CCR5 ZFN, you can see that the CD4 T cell levels actually increase much beyond what was already initially present. In addition, it's showing that the T cells which are protected from HIV infection, they expand in vivo. Same thing with this patient. You can see that the T cell levels, which were low to begin with, increased after treatment. So similarly, viral load, in both cases, the viral load is much lower in the treated as compared control. So from this part of the talk, the conclusions are that ZFN can be transiently expressed using non-integrating lentiviral vector, and HIV pseudotyping enables transduction of resting as well as activated T cells. The idea here is that you want to be able to do least of the manipulation. You take cells, T cells from HIV-infected persons, and normally what is done is you need to activate them before treating with the ZFN. In our modification, it doesn't require you to activate. You just isolate cells, treat with the lentivirus, and put them back right into the patient. So we have also been using another system, so-called the CRISPR-Cas9 system, which has been in becoming very famous in recent years because this enables easier gene editing than the ZFN that I showed you before. So what is the CRISPR-Cas9 system? Essentially, it's a bacterial adaptive immune system to defend against the foreign invaders. Essentially, you have the Cas9, this is the nucleus, and also other, for DNA recognition, an RNA molecule. In the ZF1, I told you that DNA recognition is by protein, whereas here, DNA recognition is by RNA. So essentially, in the bacteria, Cas9 protein is made, and then there are two sets of RNA. One is called the CRISPR RNA and the TRAC RNA. They are processed and finally combined, they are processed into this kind of structure, which associate with Cas9 protein and makes double-stranded DNA break. Essentially, it has two components, Cas9 nucleus and a RNA for DNA recognition. So this is the bacterial system, native CRISPR TRAC RNA duplex, whereas in for use in mammalian cells, it has been modified for this kind of structure called a single guide RNA. This is a single RNA, contains part of CRISPR RNA and TRAC RNA. So this is the part used for DNA recognition. And actually, this is how it works. Let's say this is the DNA, and this is the 5' end of single guide RNA for DNA recognition, and this is the other structure. And once this is the Cas9 protein, so this RNA binds to the Cas9 protein first and directs the Cas9 protein to the corresponding area of the DNA, where these two homology occurs. Then the nucleus domain is activated and it leads to double-stranded break in the DNA, and which is further repaired by non-homologous end joining, leading to introduction of deletions and mutations. So again, these, as with the zinc finger nucleus, the problem with this system is also the off-target effect, and off-target effect increases with nucleus exposure. Therefore, to reduce Cas9 exposure, here we use a different strategy. We use Cas9 protein pre-packed within the lentiviral vector. So here, normally, you transfect with DNA encoding Cas9 so that the DNA is continuously being transcribed and the protein is continuously being made. So to limit that exposure, we packed lentiviral particle with the Cas9, preformed Cas9 protein. So this is the strategy for that. We essentially cloned the Cas9 with the GAG pol of HIV1 and transfected cells with this Cas9 GAG pol structure and a pseudotype with VSVG. So finally, what we get is the lentiviral particle with Cas9 protein pre-packed like this. So whether it works is the question. Whether Cas9 protein is incorporated within the lentiviral vector, we can test that by looking, taking the virus and running a Western blot, plotting for P24 as well as Cas9. So both P24 and Cas9 protein are present in the lentiviral particles. Okay. So when you use this lentiviral particle to transduce TCMBL cells, which already has the gRNA in it, it leads to CCR5 knockdown. As you can, these are the controls, and the red line is the treated. And the next question is whether it works in primary T cells. Again, the same system here. We take CD4 T cells, activate with CD3, CD28 beads, and then transduce with lentivirus expressing sgRNA. And this virus expresses GFP, so sort the GFP positive cells, and then transduce them with lentivirus in which the Cas9 protein is incorporated. And a few days later, look for gene editing by cell 1 assay. So you can see here that this is the Cas9 protein encapsulated lentivirus, and this is the Cas9 encoding lentivirus. This results in long-term expression of Cas9, whereas here it is in short-term expression of Cas9. But both are equally efficient in inducing gene editing in about 20% of the cells. And the predicted off-target effect is much less when you transiently express compared to long-term expression. You can see this band here. So the system I described so far involves two steps. One is to transfect cells to make them express sgRNA, and another to transfect with the Cas9 protein encoded lentivirus. This is cumbersome. So we wanted to build a single system where we can put in both sgRNA and Cas9 protein encapsulated in the lentiviral particles. So this is what we did. We included in this expression system a lentiviral vector to express sgRNA. So it involves transduction with all these plasmids, 5 plasmids. And then finally what you get is the lentiviral particle with the Cas9 protein here and sgRNA encoding vector here. So again, if you look at the particles for P24 and Cas9, both are present. So if you look at whether it can be used for gene editing, so we have in two different cell systems here, the TCM-BL cell and Jurkat cells. And you can see that compared to control, you can induce gene editing in about 16% of these cells. And here also same thing. You are looking for CCR5 expression by flow cytometry, so it's much reduced. And this is to confirm that mutations are induced at the intended sites by sequencing. So we also use this to delete the integrated provirus from latently infected cells. This is a JLAT cell. JLAT is Jurkat cells latently infected with HIV. Normally they do not make the virus unless you activate them with TNF. Then they start making the virus, okay? So if you stain these cells for P24, initially it will be negative. Once you activate, it becomes P24 positive. So we use these cells, JLAT cells, and transduce with our lentiviral vector encoding Cas9 sgRNA. And if you look at this, after activation, this is the P24 positivity in controls, and negative cells are about 38%, whereas this is reduced to about 14% when you use Cas9 CRISPR system. And here we are using sgRNA to target LTR. So since LTR occurs at both ends of the virus, essentially if you delete the LTR, it should result in removal of the provirus. The virus should be gone. And that's what we are looking at here by looking at the GAG and POL copy numbers in these treated as compared to control cells. So essentially, in several systems, we have shown that the CRISPR-Cas9 system can be used for gene editing, and protein prepacking within the lentivirus provides a safer system for gene editing because the exposure is short. And all-in-one lentivirus makes gene editing much easier. So these are the people who did the work. Gova E, a talented postdoc, who did all the zinc finger nuclease work, and Choi and Ying Dang did the CRISPR-Cas9 knockout mice, and this is our collaborator and funding from NIH. Thank you very much.